question is a patient who works in a paint industry since 20 years came with joint pain, insomnia, fatigue, constipation, and headache. He is found to have anemia, and the enzyme most probably affected is. And the options are uroporphyrinogen 3 synthase, uroporphyrinogen 1 synthase, ALA synthase, and ferrochelator. In the description, patient works in a paint industry. So basically, this is the case of lead poisoning. And the name of enzymes which are given in the options are related to the heme synthesis. Fine. Now in the heme synthesis, there are two enzymes, particularly two enzymes which gets affected by the lead. First one is the ALA dehydratase, which is also known as the PBG synthase, which is not given in the option. And the second one is the ferrochelatase, where iron gets incorporated. Fine. So the correct answer is D. Fine. So you should remember in the heme synthesis, there are two enzymes which are inhibited by lead. First one is the ALA dehydratase and the second one is the ferrochelatase. Fine. So the correct answer is ferrochelatase. Second question is complex number 4 is inhibited by, and the options are cyanide, carbon dioxide, oligomycin and antimycin. Fine. Now this type of question is frequently asked in a various entrance examination. This particular question is from the inhibitors of electron transport chain. And the complex number 4 is inhibited by mainly three substances. Fine. It is inhibited by mainly three substances. First one is the carbon monoxide, but in the option there is a carbon dioxide, so that is not a correct answer. Second one is the hydrogen sulfide, which is also not given in the option, and the third one is the cyanide. So the correct answer is A, cyanide. So you have to remember this figure by heart because lots of question is asked from this particular topic. As you can see, complex number 4 is inhibited by three substances: hydrogen sulfide, carbon monoxide and cyanide. So the correct answer is cyanide. While oligomycin, it will inhibit the complex number 5, and the antimycin, as you can see in the picture along with the British antileviside, it will inhibit the complex number 3. Fine. So the correct answer is A, cyanide. Another question is, patient came to the hospital with abdominal pain, fatigue. Differential diagnosis is likely to be McArdle's disease. Which enzyme is affected in this patient? And the options are myophosphorylase, glucose-6-phosphatase, glycogen phosphorylase and hexokinase. The diagnosis is already given in the description, that is McArdle's disease, which is the glycogen storage disease. Fine. And basically it is the type 5 glycogen storage disease, the type 5 glycogen storage disease. And type 5 glycogen storage disease is occurs due to the deficiency of myophosphorylase. Fine, so the correct answer is A. While the second option, glucose 6 phosphatase, is related to the glycogen storage disease type 1, that is the von Gog's disease. Glycogen phosphorylase is the enzyme related to the glycogenolysis, and the hexokinase is enzyme related to the glycolysis. So over here, correct answer will be A. Fine. Another question is DNA packing is done by. Again, very simple question. And the options are A, histones, glycoprotein, nucleic acid, and helicases. The DNA packing, we all know that it is done by the histones, which are positively charged proteins. DNA are the negatively charged and histones are positively charged. And this DNA along with histone is known as the nucleosome. Fine. Why these histones are positively charged? Because they contain lysine and arginine, which are positively charged amino acids. Correct answer is A. Another question is, the following histological image is seen in the patient who is complaining of abdominal distension and growth failure, and the signs of anemia, thrombocytopenia, bone pain are present. Which enzyme is deficient in this patient? So the histological image is like this. Fine. Again, it is very commonly asked question. And the options are glucocerebrosidase, glucose-6-phosphatase, sphingomyelinase, and hexosaminidase. Fine. So this is the picture of Gaucher cells, and the Gaucher cells in the histological images appear like that of crumbled tissue paper appearance, which is also known as the wrinkled tissue paper appearance. This Gaucher cell is particularly seen in Gaucher's disease, and Gaucher's disease occurs due to the deficiency of glucocerebrosidase or beta glucosidase. So the correct answer will be A. Fine. Glucose 6 phosphatase, as we have discussed in the earlier question, it causes von Gogh disease. Sphingomyelinase deficiency occurs in Niemann-Pick disease. Hexosaminidase A is deficient in Tay-Sachs disease, while the hexosaminidase A and B is deficient in a Sandoff's disease. Fine. So that is the particular question, and the correct answer is A. Status of vitamin B2 can be determined by which of the following, and the options are hexokinase, glutathione reductase, transketolase, and thiamine. Fine, so the vitamin B2 is the riboflavin, and the active form of the riboflavin are FAD and FMN. And the level of this vitamin B2 can be determined by the level of by assessing the activity of glutathione reductase. So the correct answer will be B glutathione reductase. While the transketolase level is done for the assessing the vitamin B1 level, B1, which is also known as the thiamine. And the active form of thiamine is the TPP, and the deficiency of thiamine causes beriberi. And again, there are four types of beriberi, that is dry, wet, infantile, and cerebral. Dry beriberi will affect mostly the central nervous system, while the wet beriberi will affect the cardiovascular system. The correct answer is B glutathione reductase. Then another question is, child is brought to the hospital with complaints of abdominal pain and mental retardation. Hairy red spots are seen on the retina. What could be the diagnosis in this patient? And the options are Tay-Sachs disease, Gaucher's disease, Hunter's disease, and the Krebs disease. So the cherry red spots are mainly seen in the, as given in the option, it is mainly seen in the Tay-Sachs disease, which occurs due to the deficiency of beta-hexosaminidase A enzyme. Fine. Gaucher's disease, there is no cherry red spots are seen. Hunter's disease is the mucopolysaccharidosis 3. Again, in that cherry red spots are not seen. Krebs disease is occurred due to the deficiency of galactocerebrosidase. Fine. So the correct answer is A. Patient comes with the rice water stools. Intoxication is due to which of the following? And the options are GM1 ganglioside receptor, GM2 ganglioside receptor, GD1 alpha ganglioside receptor, and glucocerebrosidase. So the rice water stools, it suggests cholera. Cholera is caused by the Vibrio cholerae. Fine, and this Vibrio cholerae uses GM1 ganglioside receptor. So the correct answer will be fatty acid transport through mitochondrial membrane is via, and the options are carnitine, acyl carrier protein, cholesterol transferase, and lecithin cholesterol acid transferase. So the activated fatty acid transported to the inner mitochondrial membrane via carnitine, and this carnitine is synthesized with the help of two amino acids. One is the lysine and another is the methionine, and it is also known as beta hydroxy gamma trimethyl ammonium butyrate. Fine. So this is the correct answer A, carnitine, and this is the picture which shows the transport of activated fatty acid in the mitochondria with the help of carnitine, which is the step related to the beta oxidation of fatty acid. And this particular step is the rate limiting step, which is catalyzed by the CAT1, carnitine acid transferase 1 enzyme. Fine. That is the fatty acid transport. When a codon is changed to the stop codon, then it is, and the options are stop codon mutation, nonsense mutation, sense mutation, and silent mutation. So again, it is very simple question. When codon is changed to the stop codon, it is generally called as a nonsense mutation. Stop codons are three codons, that is UAA, UAA, UAG, and UGA. These are the stop codon. Fine. When codon is changed to the stop codon, it is known as the nonsense mutation. The correct answer is B. About telomers and telomerase, and the options are increased telomere length with aging, gradually decreasing length of telomers with the aging. Telomeres have coding sequences, and telomerase is found in somatic cells. So telomerase doesn't have a coding sequences, so this is not a correct answer. Telomerase is not found in somatic cells. It is found in the germline stem cells and the cancer cells. It is not seen in the somatic cells. Its length doesn't increase with aging, its length decrease with the aging. So the correct answer is B. Gradually decreasing length of telomeres with the aging. So the correct answer is B. Then Mediterranean person didn't receive Primaquine. Which pathway is affected by glucose 6-phosphate dehydrogenase enzyme deficiency? And the options are HMP pathway, glycolysis, gluconeogenesis, and Krebs cycle. So the glucose 6-phosphate dehydrogenase, it is the key enzyme of it is the key enzyme of HMP shunt pathway. Key enzyme or rate limiting enzyme of HMP shunt pathway. Fine, and this HMP shunt pathway is associated with the synthesis of NADPH and pentose sugars. Fine, it is associated with the synthesis of NADPH and pentose sugars. Fine. And this, if there is a deficiency of glucose 6-phosphate dehydrogenase, fine, if there is a deficiency of glucose 6-phosphate dehydrogenase, then we don't have to give hemaquine sulfa drugs or fawabins to the patient because it will precipitate the attack of B6PD deficiency because it will increase the oxidative stress, and as there is a deficiency of NADPH, the patient will go into the hemolytic attack. Fine. So the correct answer is A, that is HMP shunt pathway. And you have to remember this thing that glucose 6-phosphate dehydrogenase is the rate limiting enzyme of HMP shunt pathway, and it is associated with the synthesis of NADPH and pentose sugars. Then last question is woman with a history of using antidepressants present with bleeding. She gives history of bulky stools, and which of the following deficiency can lead to this? And the options are vitamin B1, B6, vitamin E, and vitamin K. So patients presents with the bleeding and she has a history of taking antidepressants. He gives history of bulky stools, that means steatorrhea, that means fat is not absorbed properly, and so we have to put our focus towards the fat soluble vitamins. So there are four fat soluble vitamins: A, D, E, and K. A and D is not given in the option, E and K is given in the option. So we have to exclude from this two that patients presents with the bleeding. So vitamin A deficiency doesn't associate it with the bleeding. Vitamin K is associated with the bleeding. And vitamin K plays important role in the gamma carboxylation of glutamic acid residue. It will convert clotting factor number 2, 7, 9, and 10 into the inactive form into the active form. Fine. So that is the post-translational modification which occurs in a clotting factors, particularly the 7, 9, and 10. Gamma carboxylation of glutamic acid residues. Fine. So the correct answer will be over here, B. So that is all about NEET PG biochemistry questions. Thank you for watching. Thank you.